Welcome to my YouTube channel. So here today we are going to be looking at HIV. What are the series of steps and the differences between HIV and the lytic cycle? So to begin with, HIV is made up of RNA. So now, why is this important? We are going to be looking at something that we are going to call reverse transcriptase and its role. So to begin with, before we look at HIV, how the cycle is, let's look at some of the characteristics. So now, when we are looking at HIV, the general characteristics are that we first need to know what HIV stands for. So HIV stands for Human Immunodeficiency Syndrome. So now, what are some of the other things? So HIV is spread through sexual activities. So it is spread through sexual activities. So HIV also attacks what we are going to be calling the CD4. So CD4, in case they bring multiple choice and they put the full meaning, you should know that it is a cluster of differentiation. So a cluster of differentiation 4. So that is CD4. So you attach 4. So that is it attacks the CD4. So CD4 are basically what we call the receptors. They are CD4 helper T cells, like that. T helper cells. Let's begin with T. So that is one of the things that we need to know. So HIV is a retrovirus. So HIV is a retrovirus. What does this mean? So it means to say it is capable of reversing DNA to RNA or vice versa. So that's basically it about HIV. Now, this is the general background of HIV before we look at the cycle. Now, the symptoms. What are some of the symptoms? The symptoms of HIV is night sweats. So we have night sweats. We also have swollen lymph nodes. Now, if you recall what lymph nodes are from your grade 12, lymph nodes are where most of the red, not the red, but the white blood cells are produced. So if, let's say, this is the neck, we have the thoracic lymph node here where they are produced. We might have the axillary, which occurs in the armpit. So at this point, if you have a swollen lymph node, that is one of the signs of HIV. So this is the head of someone. Hope you are able to see that this is the head of someone. It's just bald. So we have, this is the head of someone. So the thoracic lymph node is found on the neck. So swollen lymph node is one of the signs and symptoms. We also have what we are going to call the mouth ulcers. We have mouth ulcers or a white tongue. So now, the other series of events, before we look at that, we can also have the muscle aches. So these muscle aches are pains and also just generally fatigue, being tired even though you are not doing any work. So those are some of the symptoms. Now when we look at the cycle, so remember, don't forget what we looked at. So all the terms that I'm going to be mentioning, they are going to be very, very important. So the first thing, these are the terms that you're going to be using. Number one, you're not going to use binding. Binding is okay, but I prefer that you use, the first stage is attachment. So when so as you can see, this is the virus. So this is our our virus here. So now this virus attaches to the receptor. So whatever is like this is the receptor. Now this receptor is what we are going to call the CD4. So the cluster of differentiation. So now sometimes they might not just attach to the CD4, but they can attach to other receptors. So this is the receptor, which is the CD4. They can also attach to helper receptors, which we are going to CCR5. So sometimes they might ask you, especially for multiple choice, we can have CCR5. We can also have the CXCR4. So, but generally, the most important thing is the CD4. So they attach to the CD4. They can also attach to other core receptors such as the CCR5 and the CXCR4. So now when we look at after there is attachment, which in brackets, when we look at attachment, which in brackets, we are going to have binding. So when they bind using their receptors, but let's look at the material that makes up the these protein coats. If you remember, these are coats, the protein coats or spikes. So these spikes, GP120. So GP120 are the spikes that are found on the just these same green protein coats that you can find. So these are called GP. So the GP120 is found on the virus. So the GP120. So what does GP mean? It means the glycoprotein. Glycoprotein is a glucose bonded to, so glucose bonded to protein. So it is made up of glucose and proteins. That's the GP120. So these are the spikes. So they attach to the CD4 and other core receptors such as CCR5 and CXCR4. So generally we are going to be interested in the CD4 like that. Then when we look at the next stage, when it binds like that, it is now going to undergo the process which we are going to call fusion. Fusion, in other words, we are going to call it penetration. So penetration, fusion, or entry. So in this case, let's go for penetration. In brackets, we can have fusion. So under this one, what begins to happen is that the HIV genome, which is the genetic material, is injected into the cell. So it injects the cell. Now remember, we said that DNA is RNA, like that. It is made up of, not DNA, sorry. The HIV genetic material is made up of RNA. So as you can see here, we have this is our RNA, like that, our RNA, we have our RNA like that. Then this material, through the process of reverse transcription, will be able to convert. So under reverse transcription, this is done with the help of the enzyme called reverse transcriptase. So reverse transcriptase will be able to convert the HIV RNA to DNA, like that. So when it converts it to DNA, the next stage will be integration. So integration is where now we are going to have the DNA enters the nucleus. So we might have, so this is the cell. If we have the nucleus like that, the DNA will be able to enter the nucleus. When it enters the nucleus, so as you can see, it will be able to integrate itself like that. So it will be able to integrate itself, or rather insert itself into the host DNA, as you have seen this red thing. So we have something like this. Then it is able to more like attach itself or insert itself to the host DNA, like that. So now what is formed after integration? So this whole thing is what we are going to call, the integrated form is what we are going to call the provirus. So you can just be able to think of, if you are a pro, you are able to enter certain environments that are not even yours. So it is even called a provirus, like that. So that is basically what integration is all about. Then after that, so due to the process in which there is the formation of the provirus, what begins to happen is that the host machinery is hijacked, meaning to say it has been taken over. So immediately the DNA attaches its now DNA, the HIV attaches its now made DNA to that. It hijacks the host material, the host cell machinery, should I say. So this machinery is the one that makes, so as you can see, this border thing, this is the nucleus. So now when it hijacks like that, it is now able to use this to create certain proteins and RNA. So when the RNA is made, as you can see, we have this yellow substance. So this is RNA. As you can see from the beginning, this yellow thing is RNA. So this is RNA. Even here, what is made is RNA, because DNA is made up of RNA and other proteins like this. So these proteins will now be able to be put in this thing, then it will be able to close up, and therefore there will be the full creation of an HIV virus, like that. So this the last process that we are going to be looking at after we have replication. So there is the making of proteins, there is the making of proteins and RNA, which are deposited at the cell surface, like that. Then finally we have assembly. Assembly is the process by which the viral RNA and proteins come together at the cell membrane. So when they come together at the cell membrane and everything is done, that is what we call assembly. So when they assemble now, they will now be able to undergo the process of release, like that. So basically these are the steps. So we have attachment, we also have penetration as the second stage, reverse transcription, where the DNA RNA is the HIV RNA is converted to DNA. Then there is integration, in which the DNA enters the nucleus and is inserted in the host's DNA genetic material. Then we have replication. Under replication, there is the formation of proteins as well as the RNA. So remember, so the DNA is using reverse transcriptase to also be able to convert the DNA that it made to a form which is RNA, which makes up the virus. As they move to the surface before release, we are going to have what we are going to call assembly, in which the proteins and assembly. So under this process, what will happen is that the material will be able to go to the cell surface, and when it goes to the cell surface, what begins to happen is that there will be the assembly of proteins as well as the genetic material. So when that happens, that simply means to say they can now be released. So that is what the cycle of DNA is all about. So that is the cycle of DNA. Now let's just look at the last thing, which is the difference HIV cycle and the lytic cycle. What are the differences? So the differences are, to begin with, When we look at number 1, we are going to have the lytic cycle does not involve reverse transcriptase. So that's the first difference. So just as we have said, the lytic cycle does not involve reverse transcriptase. So if you recall from the video of the lysogenic and the lytic cycle, you will find that there was no mention of the enzyme reverse transcriptase, while HIV does have the reverse transcriptase. The other thing that is important is that the lytic cycle does not involve integration. Under the lytic cycle, there is nothing like the DNA, there's nothing like the DNA implants itself in the host DNA. So that is what we basically mean by the lytic cycle does not involve integration, while HIV cycle involves integration of the viral genome into the host cell's DNA. The number 3, we are going to have the lytic cycle is rapid and causes, or rather leads, this is the word, leads to destruction. It leads to the destruction of the host cell. While HIV cycle can be slow, sometimes you might think that you might not have HIV, and may remain latent, meaning to say inactive, without immediately killing the host. So latent is more like the delayed period, latent without immediately killing the host. Last difference is that under the lytic cycle, we have it produces more new viruses, because it is very fast, viruses quickly, and causes cell lysis, which is the breaking of the cell. While HIV cycle may delay virus production, so it may take time and also persist inside the host for a very long time without breaking it, though there comes a time when there shall be cell lysis like that. Let's see other diagrams that we have. Yeah, so attachment, we have fusion, then we are going to have reverse transcriptase. Then from reverse transcriptase there will be integration. It's more like this what this is, put the virus is attached now here. So the virus came, as you can see, it came as RNA, then it is converted into DNA. From DNA it is now integrated in the genetic material of the cell. Then this will now be able to, after the replication process, it will now be able to be assembled, where the proteins and the new genetic material, the new RNA which is made now, will be able to enter the cell like that. Then after that we are going to have release after maturation like that. So that is basically the process of HIV, the HIV cycle. So this is it, and if you have any question, just comment in the comment section. See you in the next tutorial.